Semaglutide was doing something in the brain. They found that levels of p-tau, a protein in the brain that marks Alzheimer's disease, it went down. Markers of neuroinflammation went down. A protein neurogranin, which reflects how well synapses, remember those connections between brain cells, how well they're functioning, that went down. These are not trivial measurements. These are the same biomarkers that anti-amyloid drugs move when they're working in research studies. So the drug was engaging its biological targets and having a measurable effect, but it just wasn't helping patients clinically. How do we explain that? Why would a drug produce biological changes in the brain and still fail to slow the disease process? The answer almost certainly is scale and timing. Did you get enough drug into the brain, and did you give it early enough for it to have an effect? Let me give you an example. Let's say you're imagining a house that has been quietly filling with moisture for 20 years. The damage to the house's structure, corrosion in the pipes, deterioration in the structure, that's been accumulating for decades. Now, imagine you walk in, open the window, let fresh air in, reduce the moisture level drops. That's real, that's measurable, but the structural damage doesn't reverse because you cracked one window open and reduced the moisture at that time. The damage has been done. That's likely what's happening in this situation.